Basics of Medical Biochemistry. First edition, 2022. Chapter 3. Chemistry of Carbohydrates, Part 1 of 2. By Professor Dr. Muhammad Jawad. MBBS, Dow, M.Phil Philosophy, Ph.D., Dip M.E. Head of the Biochemistry Department-Fazaia Ruth Pfau Medical College-PAF Base Faisal-Karachi-Pakistan. Ex-Professor of Biochemistry, Qassim University Kingdom of Saudi Arabia. Ex-Professor, Head of the Biochemistry Department and Vice Principal. Liaquat College of Medicine and Dentistry and Fatima Jinnah Dental College, Karachi, Pakistan. Chapter 3. Chemistry of Carbohydrates. 3.1 Introduction. Carbohydrates are defined as polyhydroxyaldehydes or polyhydroxyketones or the compounds which give them upon hydrolysis. Basic concepts. Carbohydrates are widespread in nature both in plant and animal kingdoms. They are a major source of energy in most organisms. They have a wide range of functions. They also play an important role in lubrication, cellular intercommunication, and immunity. They are hydrates of carbon. They are compounds of carbon, hydrogen, and oxygen. The ratio of CHO equals 1:2:1. General formula is CH2O, N. They are also known as saccharides, derived from saccharin, Greek, means sweet. Commonly known as sugars. Their caloric value is 4.1 kcal/g. Most abundant organic compounds in nature. They are the chief constituent of our diet and act as an instant source of energy for the body. The main structural component of the living system. Absorbable forms are glucose, fructose, and galactose. The extra amount of carbohydrates is stored in the liver and muscles while the rest is converted to fats and proteins. The requirement in a balanced diet is 50 to 70% and the carbohydrate reserve in a normal adult is about 500 grams. 3.2 Classification of carbohydrates. Carbohydrates are classified into 5 major groups. 1 Monosaccharides, 2 Disaccharides, 3 Oligosaccharides, 4 Polysaccharides, 5 Derived carbohydrates. 3.2.1 Monosaccharides, these are the simplest carbohydrates. They cannot be hydrolyzed further. Monosaccharides are colorless and crystalline substances with a sweet taste. They are soluble in water. Monosaccharides are simple sugars having a free aldehyde or ketone group. Glucose, aldehexose, and fructose, a ketohexose, are the most common monosaccharides. Their features are the simplest form of sugars, absorbable form of sugars, fully reactive form of sugars, the most stable form of sugars. General formula equals CnH2n. Further classified based on the number of carbon atoms as trioses, tetrases, pentoses, etc., and based on functional group as aldoses or ketoses. Table 3.1 Types of monosaccharides. No of C. Atoms. Generic name. Systemic names. Examples. 3. Triose, aldotriose, glyceraldehyde, ketotriose, dihydroxyacetone. 4. Tetras, aldodetras, erythrose, ketodetras, erythrulose. 5. Pentose, aldopentose, ribose, ketopentose, ribulose. 6. Hexose, aldohexose, glucose, ketohexose, fructose. 7. Heptose, aldoheptose, glucoheptose, ketoheptose, sidoheptulose. 3.2.2 Disaccharides. Disaccharides are formed by the condensation of 2 monosaccharide molecules. The monosaccharides are linked together by glycosidic linkages. General formula equals C12H22O11. Easily hydrolyzed by acid, alkali, or enzymes. Important examples are maltose equals glucose plus glucose, fruit sugar. Lactose equals glucose plus galactose, milk sugar. Sucrose equals glucose plus fructose, table sugar. Isomaltose equals glucose dimer having, 1 rightward arrow 6, glycosidic linkage. Trehalose equals glucose dimer having, 1 rightward arrow 1, glycosidic linkage. Cellubiose equals glucose dimer having, 1 rightward arrow 4, glycosidic linkage. 3.2.3 Oligosaccharides, they are formed by the union of 3 to 10 units of monosaccharides. These monosaccharides are joined together by O-glycosidic linkage. Important examples are maltotriose, composed of 3 glucose units, dextrin or limit dextrin, a short-chained branched amylopectin remnant of about 8 to 10 glucose units, produced by hydrolysis of amylopectin with alpha amylase, raffinose or sugar beets, polymer of galactose, glucose, and fructose. Some oligosaccharides may have amino groups or N-acetylated amino groups. 3.2.4 Polysaccharides, they are formed by the condensation of a large number of monosaccharide molecules, more than 10 units. They have high molecular weights and are only sparingly soluble in cold water. They form a colloidal solution when heated with water. They are not sweet and do not exhibit any of the properties of the aldehyde or ketone group as in the case of monosaccharides and disaccharides. They serve as a store of fuel and also worked as a structural component of cells. They are generally divided into following 2 main groups. 1. Homopolysaccharides. These polysaccharides are made up of the same kind of monosaccharide units, example starch, glycogen, cellulose, dextrins, polymers of glucose, and inulin, polymer of D-fructose. 2. Heteropolysaccharides. These polysaccharides are made up of different kinds of monosaccharide units. Depending on animal or plant origin, they have been further subdivided into 2 groups. Animal origin. Most common example is the mucopolysaccharide group, which includes hyaluronic acid, heparin, chondroitin sulfate, blood group polysaccharides and serum mucoid. In addition to carbohydrate parts, acid groups are also present in them. Another example is glycoconjugates, which include proteoglycans, glycoproteins, and glycolipids. 2. Plant origin. Most common example is the mucilage group, which includes agar, vegetable gums, and pectins. In plant cell walls, cellulose occurs in densely packed fibrils, called hemicellulose. The most common polysaccharides are starch, glycogen, and cellulose. Starch is the storage form of glucose in plants. Glycogen is the storage form of glucose in animals. Cellulose is a structural polysaccharide of plant cells. Although it is present in the human diet, but it cannot be digested due to a lack of enzyme cellulase. 3.2.5 Derived carbohydrates. These are obtained from the above carbohydrates as a result of various chemical reactions upon them. Like oxidation products as various sugar acids such as gluconic acid, glucuronic acid and glucuric acid. Reduction products example vitamin C, polyhydroxy alcohols example glycerol, amino sugars example glucosamine, deoxy sugars example 2-deoxyribose of deoxyribonucleic acid. 3.3 Chemical types of carbohydrates. Sugars can be divided into 2 groups. 1. Reducing sugars. Reducing sugars are those which have potentially active aldehyde or ketone groups in their structure. In other words, these sugars have free anomeric carbon atoms. The sugars that have a free sugar group are reducing sugars. All monosaccharides such as glucose, fructose, and galactose, etc., are reducing sugars because they have a free anomeric carbon atom in their structure. Disaccharides such as maltose and lactose are also reducing sugars because they possess a potentially active aldehyde group. 2. Non-reducing sugars. Their anomeric carbon atoms are engaged in making bonds with each other, and these are the sugars that do not have free aldehydic or ketonic groups. Thus, they cannot reduce other substances. Some disaccharides such as sucrose and all polysaccharides are non-reducing sugars. However, on hydrolysis with acid, base, or enzymes they yield smaller subunits that have reducing properties. The asymmetric, chiral, carbon atom, penultimate carbon atom and reference sugar, a carbon atom to which 4 different atoms/groups of atoms are attached or when all the valences of C are filled by different groups. Figure 3.1 Asymmetric carbon atom. Glyceraldehyde, glycerose, is the smallest sugar having 3 carbon atoms, can be represented in the following form as Fischer projection or open chain formulas. Carbon number 2 is an asymmetric carbon atom in both figures. In the figure on left, the O group is attached to the right side of an asymmetric carbon atom and this structure is called diglyceraldehyde, while in the figure on right the O group is attached to the left side of the asymmetric carbon atom and this structure is called as L-glyceraldehyde. Cho-cho. HCO-CH. CH2O-CH2O. D-glyceraldehyde, L-glyceraldehyde. Figure 3.2 D and L type of glyceraldehyde. The nature of the D or L form of all those monosaccharides having more than one asymmetric carbon atom is decided by considering the position of the O group around the penultimate carbon atom. It is the asymmetric carbon atom that is situated farthest from the potential aldehyde or ketone group and it has the highest number in the carbon chain. Those monosaccharides whose penultimate carbon atom has the same configuration as the asymmetric carbon atom of D-glyceraldehyde are called D-monosaccharides. As all the monosaccharides are compared with D and L-glyceraldehyde for the conformation of their D or L form, the glyceraldehyde is known as reference sugar. H1C equals O, H1C equals O. H2CO, Ho2CH. Ho3CH, H3CO. H4CO, Ho4CH. H5CO, Ho5CH. 6CH2O, 6CH2O. D-glucose, L-glucose. Figure 3.3 D and L-glucose, Fischer's projection. The biological and optical activity of monosaccharides is dependent on the number of asymmetric carbon atoms. All monosaccharides must have at least one asymmetric carbon atom except for dihydroxyacetone, which does not possess an asymmetric carbon atom, that's why it is biologically inactive. 3.4 Isomerism in monosaccharides. Isomers are compounds having the same chemical formula but differ in their structural formula. For example, glucose, fructose, mannose, and galactose have the same chemical formula of C6H12O6 but their structural formula is different from one another, so they are isomers of each other. The following important types of isomerism are seen in monosaccharides. 3.4.1 Stereoisomerism. The presence of asymmetric carbon atoms provides the basis of isomerism in a compound. Stereoisomerism means 2 or more monosaccharides having the same chemical composition but differing from each other regarding the configuration, i.e., the arrangement of their atoms in space is called stereoisomerism. 3.4.2 Enantiomers. The pair of stereoisomers that are mirror images of each other around asymmetric carbon atoms are called enantiomers. For example, the D and L series of monosaccharides. The orientation of H and O groups around the highest, penultimate, carbon atom determines the series. D and L monosaccharides are metabolized by specific enzymes. D type of monosaccharides is common in mammalian tissues. The number of possible isomers of a monosaccharide can be predicted by Van 't Hoff's rule equals 2n, where n represents the total number of asymmetric carbon atoms in a compound. According to this rule, number of possible isomers of glucose in the chain form is 2 times 2 times 2 times 2 equals 16, as it has 4 asymmetric carbon atoms, i.e., carbon number 2 to 5. But in the case of the cyclic form of glucose, the number of possible isomers increases up to 32 because of the conversion of carbon number 1 into an asymmetric carbon atom as a result of cyclization. 3.4.3 Enomers. These isomers differ from one another in configuration around an enomeric carbon atom. It is carbon number 1 in aldoses and carbon number 2 in ketoses. These two types of isomers are called N-beta enomers. These do not mirror images of one another. These are called diastereoisomers. 3.4.4 Epimers. These isomers differ in configuration around one specific carbon atom. For example, glucose and galactose differ from each other only in the position of the O group around carbon number 4, so they are called C4 epimers. Similarly, mannose is a C2 epimer of glucose. It is to be noted that galactose and mannose are not epimers of one another. H1C equals O, H1C equals O, H1C equals O. H2CO, H2CO, Ho2CH. Ho3CH, Ho3CH, Ho3CH. H4CO, Ho4CH, H4CO. H5CO, H5CO, H5CO. 6CH2O, 6CH2O, 6CH2O. D-glucose, D-galactose, D-mannose. Figure 3.4 epimers, chain form of glucose, galactose, and mannose. 3.4.5 Optical isomerism, the ability of biomolecules to rotate the plane of polarized light either on the left or right-hand side of a monochromatic beam. When passed through a sugar solution, it is rotated either clockwise or anti-clockwise. They all are stereoisomers as well as enantiomers and may be epimers too. They exhibit optical activity by their asymmetric carbon atom. There are two types of optical isomers. I-dextrorotatory, which rotates light to right. Usually, D-isomers are dextrorotatory and they are denoted by D or plus. 2-level rotatory, which rotates light to left. Usually, L-isomers are level rotatory and are denoted by L or. But it is not compulsory. For example, D-fructose is level rotatory, although it is a D-monosaccharide. The rotation of plane polarized light by biomolecule is determined by a polarimeter. They have specific rotations, for example plus 140 for D-glyceraldehyde, 140 for L-glyceraldehyde, plus 52.7 for D-glucose and 52.7 for L-glucose. 3.4.6 Pyranose-furanose isomerism. Different monosaccharides may exist in a cyclic form as pyran, 6-membered ring, or furan, 5-membered ring. 3.5 Cyclization of monosaccharide and ring structure of glucose. D-glucose is a type of aldehexose. Its open-chain straight structure, as suggested by Fischer in 1891, mentioned that it has 4 asymmetric carbon atoms, i.e., carbon number 2 to carbon number 5. According to Van 't Hoff's rule, total numbers of possible isomers of glucose are 16. But in fact, there are 32 isomers possible for D-glucose in an aqueous medium after its cyclization. During cyclization, carbon number 1 and carbon number 5 react together and bound through an oxygen bridge. As a result, the chain form of glucose is converted to cyclic form with the appearance of one more asymmetric carbon atom, i.e., carbon number 1 becomes a new asymmetric carbon atom called an anomeric carbon atom. There are 2 enomers of D-glucose as D-glucose and D-glucose depending upon the position of the O group around the anomeric carbon atom as below and above the plane of the ring respectively. The alpha and beta forms of glucose that occur in cyclic form do not show the aldehyde group as a functional group of glucose. During cyclization of hexoses, there occurs the formation of hemiacetyl or hemiketyl through condensation of aldehyde or ketone group with the hydroxyl group of the same compound in the ratio of 1:1. As a result of this condensation, the carbon atom of aldehexose or ketohexose is called hemiacetyl or hemiketyl carbon respectively. This carbon is a newly formed asymmetric carbon already termed an anomeric carbon atom. Figure 3.5 Cyclic and chain forms of D-glucose. 3.6 Muta-Rotation, muta equals change. When a freshly prepared solution of D-glucose is observed in a polarimeter, it is seen that its specific rotation initially keeps on changing and then becomes fixed after some time. This phenomenon is called muta-rotation. The property of muta-rotation in glucose is because on being dissolved in water, the glucose molecules go on changing from one form to another form. At equilibrium, about 38% of D-glucose has a specific rotation at plus 112 while 62% of it has a specific rotation at plus 19. These two types of glucose are termed as N form of D-glucose. After a definite time, the optical rotation is fixed at plus 52.7. Figure 3.6 Muta-Rotation in glucose. The rotation of plane polarized is not being observed in the following conditions. 1 when there is no asymmetric carbon atom in a compound. For example, dihydroxyacetone does not exhibit optical rotation as it does not possess any asymmetric carbon atom in its structure. 2 when equal amounts of dextrorotatory and levorotatory compounds are present in the mixture. In this case, the two isomers perform optical rotation in opposite directions with equal force. That's why no net rotation is observed. This is known as external compensation, and such a mixture is called a racemic mixture. 3 when mixture contains mesocompound. There is a phenomenon of internal compensation because the molecule has two halves, one half rotating light to the right and another half to the left. As a result, no net movement is observed, although the compound possesses an asymmetric carbon atom. An example of mesocompound is mesotartaric acid. Haworth's formula of glucose, pyrin, is a 6-member ring with 5 carbons and 1 oxygen atom. It is a ring formed between carbon number 1 and 5 of glucose as a result of intramolecular hemiacetal formation to produce pyranose. The pyranose form of glucose was first given by Haworth. Another aldehexose also has a pyranose ring in its structures. In these types of structures, heavier bonds are placed towards the front of the figure. There is a different configuration of H and O around anomeric carbon atoms to produce enomers. Aldehexoses may also exist as furanose, which is a derivative of a furan. Furan is a 5-member ring with 4 carbons and 1 oxygen atom. In this structure, the ring is formed between carbon number 1 and 4. The common and stable form of aldehexose is aldopyranose than aldofuranose. In ketohexose, the hemiketal linkage is produced by the reaction between the ketone group at carbon number 2 and the O group on carbon number 6 or 5 to produce pyranose or furanose respectively. Just like glucose, there are 2 enomers of fructose as alpha-D-fructopyranose and beta-D-fructopyranose, 4 6-membered pyranoses, and alpha-D-fructofuranose and beta-D-fructofuranose, 4 5-membered furanoses. Fisher and Haworth's formula of glucose is still unable to explain the true conformation features of this sugar. Figure 3.7 Furan, pyrin, and their derivatives. The chair and boat forms of pyranose ring of glucose have been invented to give more scientific conformational aspects of glucose and other monosaccharides. The chair form is found to be more stable than the boat form of alpha-D-glucose. Figure 3.8 Boat and chair conformation of alpha-D-glucose. 3.7 Some important monosaccharides. 3.7.1 Hexoses, all those simple sugars, monosaccharides, which contain 6 carbon atoms per molecule are called hexoses. Hexoses may be classified based on their functional group. They may be aldehexose if the aldehyde group is attached at position 1, or they may be ketohexose if the ketone group is attached at position 2. Important examples include glucose, fructose, galactose, and mannose. 3.7.1.1 Glucose, it is an aldehexose also known as dextrose or blood sugar. It is found as free monosaccharide in fruits and vegetables of many plants, so commonly known as grape sugar. It also occurs in combined form as glycosides, disaccharides, oligosaccharides, and polysaccharides. It is the chief source of energy for all living organisms. Structure: the chemical formula of glucose is C6H12O6. It contains 6 carbon atoms. It has the functional group an aldehyde group and is therefore termed aldehexose. Glucose, cyclic form, glucose, chain form. Figure 3.9 Structure of glucose. Characteristic features of glucose. It is produced in the body by the hydrolysis of starch, sucrose/table sugar, cane sugar, maltose, lactose, and carbohydrates of grains and cereals. Glucose is a primary source of energy for brain cells and erythrocytes. The ultimate and final product of all types of carbohydrates in the body is glucose. It is regarded as major metabolic fuel of the body and acts as an instant source of energy for cellular functions, like muscle contraction, neural conduction, tissue repair, etc. It is absorbed in the small intestine via the portal vein and passed through the liver, where the extra amount is converted into glycogen by the process of glycogenesis. Normal fasting glucose level is 70-110 mg/dl or mg% and random blood glucose level is 80-160 mg/dl or mg%. Normally, a very less almost negligible amount of glucose is passed in the urine, which is not detectable by usual laboratory procedures. The glucose appears in the urine only when the blood glucose level crosses the renal threshold level, which is 180 mg/dl. The glucose is not utilized by the tissue until phosphorylated by ADP. The glucose exhibits almost all properties of monosaccharides like good reducing agent, oxidized to sugar acid, reduced to alcohol, ester formation. With phenylhydrazine, it forms a needle-shaped yellow-colored crystal of ozazone. Sweet and water-soluble, 74% as sweet as sucrose, etc. Glucose can be converted into non-carbohydrate substances, example fat, and in this way regulate overall metabolism. Glucose may be synthesized from non-carbohydrate residue within the body via gluconeogenesis. Clinical aspect: blood sugar concentration refers to the amount of glucose in the blood. Failure to maintain blood glucose within the normal ranges leads to hypoglycemia equals low blood glucose levels. Hyperglycemia equals high blood glucose levels. Persistent hyperglycemia is characteristic of diabetes mellitus, in which there is a failure of blood sugar regulation. The appearance of glucose in the urine is called glycosuria. Hyperglycemia and glycosuria are the two important features of diabetes mellitus. Diabetes mellitus. Diabetes mellitus is a clinical syndrome characterized by disturbance in carbohydrate, lipid, and protein metabolism. The disease may result from a defect in insulin secretion, insulin action, resistance, or both. Hormonal regulation of blood glucose. Two mutually antagonistic metabolic hormones are responsible for keeping the concentration of glucose in blood within the normal range. These are insulin, decreases blood glucose, and glucagon, increases blood glucose. The glucose given to the patient intravenously in the form of drip is dextrose, having a concentration of 5% to 10% usually. Glycemic index. The increase in blood glucose after a test dose of carbohydrate compared with that after an equivalent amount of glucose is known as the glycemic index. It is the ratio of the area under the blood glucose curve after eating a particular food divided by the area after taking the same amount of carbohydrate as glucose. Foods with a low glycemic index generally aid good metabolic control since they cause less fluctuation in insulin secretion. Carbohydrate counting. It is the relationship between the dose of insulin required and the amount of carbohydrate taken in the diet. In this regard, diabetic patients are instructed to administer 1 unit of regular insulin or insulin lispro for each 10 to 15 grams of carbohydrate eaten at a meal. 3.7.1.2 Fructose. It is a ketohexose or levulose commonly known as fruit sugar because of its high concentration in fruits. It is an important member of the ketose family. Fructose is twice as sweet as sucrose, hence it is used in much smaller amounts as compared to sucrose. Sources: sugarcane, honey, fruits like berries, melons, and some root vegetables like beets, sweet potatoes, onions, etc. The chief dietary source is sucrose, taken as table sugar. Structure: fructose is a reducing monosaccharide with a ketone group as a functional group. Fructose is an isomer of glucose with the same chemical formula, C6H12O6, but has a different structural formula. Fructose, cyclic form, fructose, chain form. Figure 3.10 Structure of fructose. Characteristics: features of fructose. In the small intestine, it is produced by the action of sucrase on sucrose, absorbed by the portal vein to the liver where it is isomerized to glucose for use in the body. It can be converted into glucose in the liver, kidney, and small intestine. It is rapidly catabolized as compared to glucose and is a good source of energy. Excess dietary fructose is harmful as it leads to increased synthesis of triglycerides. In the food industry, fructose is used as a sweetening agent in processed food products. The fructose found in the human body is levorotatory and readily converted to glucose. It is the sweetest of all sugar, being 173% as sweet as sucrose. A significant amount of fructose is found in the seminal fluid, where it is regarded as a primary source of energy for the maturation of spermatozoa. It exhibits almost all properties of monosaccharides like good reducing agent, dehydrated by strong acid, reduced to alcohol, oxidized to sugar acid. With phenylhydrazine, it forms the same type of ozone crystals as glucose. Fermented by yeast. In the laboratory, the only test to differentiate it from glucose is Seliwanoff's test, which is positive for all sugar-containing ketone groups. Clinical aspect. Clinical disorders related to fructose metabolism can result from excessive fructose consumption that exceeds the body's ability to efficiently convert sugar into metabolic intermediates. Essential fructosuria. It is a benign asymptomatic condition due to the deficiency of the enzyme fructokinase, resulting in the accumulation of fructose in the blood beyond the normal range and appearing in the urine. Hereditary fructose intolerance. The body cannot digest fructose due to the deficiency of the enzyme aldolase B. This results in the accumulation of fructose in the liver, kidney, and small intestine. Signs and symptoms of this disorder are severe hypoglycemia, vomiting, jaundice, sweating, tremors, hemorrhage. Treatment is rapid detection and removal of fructose and sucrose from the diet. 3.7.1.3 Galactose. It is aldehexose. Galactose is a type of sugar that is less sweet than glucose. It is considered a nutritive sweetener because it has less food energy. Sources. It is found in dairy products, sugar beets, and other gums and mucilage. It is also synthesized in the body, where it forms a part of glycolipids and glycoproteins in several tissues. Structure. It is an epimer of glucose that differs in configuration at carbon number 4. Galactose, cyclic form, galactose, chain form. Figure 3.11 Structure of galactose. Characteristic features of galactose. Galactose is an aldehexose having the same empirical formula as glucose, i.e., C6H12O6. Galactose is required to synthesize a variety of biomolecules. It doesn't occur free in nature. In the body, it is found as the constituent of milk, in the form of lactose in combination with glucose, and in cerebrocytes, the glycolipids of nervous tissues. As a polysaccharide, it is found as the galactan in mosses and seaweeds, etc., also found in seed coats of legumes. It is produced in the small intestine by the action of lactase on lactose absorbed via the portal vein and converted to glycogen in the liver. It exhibits most of the properties of monosaccharides like good reducing agent, dehydrated by strong acid, oxidized to sugar acid, reduced to alcohol. With phenylhydrazine, it forms the rod-shaped crystals of ozone. Fermented by yeast. Clinical aspect. Galactosemia is an inherited disorder representing the raised level of galactose in the blood. It is an inborn error of galactose metabolism due to the deficiency of enzymes required for the conversion of galactose into glucose. In this condition, galactose also appears in the urine. Initially, newborns are symptom-free. The symptoms that appeared on the third day of life are vomiting and jaundice. After 2 to 3 months, symptoms associated with galactosemia become more severe and include liver disorders, cataract formation, mental retardation, and severe diarrhea. It is treated by lactose and galactose-free diet. Some ongoing research suggests that galactose may have a role in the treatment of focal segmental glomerulosclerosis, a kidney disease resulting in kidney failure and proteinuria. 3.7.1.4 Mannose. Mannose is C2 epimer of glucose. On ingestion, it is absorbed as such and converted into glucose. It is not well metabolized by humans. It is present in numerous glycoconjugates including N-linked glycosylated proteins. Most of its chemical reactions are similar to glucose. 3.7.2 Pentoses. pentoses are monosaccharides with 5 carbon atoms. They either have an aldehyde functional group at position 1, aldopentose, or a ketone functional group at position 2, ketopentose. Pentoses are well distributed in nature both in the plant and animal kingdom. Important examples are ribulose, xylulose, ribose, xylose, deoxyribose, fucose, etc. The aldopentoses have 3 chiral centers, asymmetric carbon atoms, and 8 different stereoisomers, those isomers that have the same molecular formula and sequence of bonded atoms, but differ only in the 3-dimensional orientations of their atoms in space, are possible. The ketopentoses have 2 chiral centers and 4 different stereoisomers. Pentoses are good reducing agents, converted to respective furfuryls by acids, and can form ozazone crystals with phenylhydrazine but cannot be fermented by yeast. 3.7.2.1 Ribose and deoxyribose. Ribose is an aldopentose. It contains 5 carbon atoms, which in their acyclic form, have an aldehyde functional group at one end. Typically, this species exists in the cyclic form. The ring form of ribose follows a similar pattern as that for glucose with one exception. Since ribose has an aldehyde functional group, the ring closure occurs at carbon number 1, which is the same as glucose. The exception is that ribose is a pentose, therefore a 5-membered ring is formed. The O on carbon number 4 is converted into the ether linkage to close the ring with carbon number 1. This makes a 5-membered ring having 4 carbon atoms and 1 atom of oxygen. Figure 3.12 Structure of pentoses, ribose, left, and deoxyribose, right. Deoxyribose. Deoxyribose is also an aldopentose. It is exactly similar to ribose, except in one place. The only difference between ribose and deoxyribose is that at carbon number 2 instead of the hydroxyl group only a hydrogen atom is present. Characteristic features of pentoses. Ribose is one of the 3 chief constituents of RNA. Deoxyribose is one of the 3 chief constituents of DNA. L-xylulose is excreted in the urine in essential pentosuria, a benign autosomal recessive disorder due to the deficiency of L-xylulose reductase. L-xylulose is found as an intermediate product in the uronic acid pathway of glucose metabolism. Ribulose occurs in phosphorylated form as an intermediate in the pentose phosphate pathway. D-Glucose forms a constituent of lixoflavin isolated from human heart muscle whose function is not clear. Pentoses are also found in many important substances like ADP, GTP, CTP, UTP, NAD+, FMN, FAD and coenzyme A. 3.7.3 Tetrases. The monosaccharides contain 4 carbon atoms in their primary chain are called tetras. Important examples are erythrose, erythrulose and threose. Tetrases may be aldotetrous or ketotetrous depending upon whether the functional group is aldehyde or ketone respectively. 3.7.3.1 Erythrose. It is an aldotetrous with the chemical formula C4H8O4, the same as threose. The only difference between erythrose and threose is depicted in the diagram below. 3.7.3.2 Threose. Threose is a tetras carbohydrate with the chemical formula C4H8O4. It has one aldehyde group and belongs to the aldose family. It exists both in D and L stereoisomers. 3.7.3.3 Erythrulose. It is a ketotetrous. The structure of both L and D stereoisomers is given in the following diagram. Figure 3.13 Structure of tetrases. Characteristic features of tetrases. Erythrose 4-phosphate, a phosphorylated derivative of erythrose, serves as an important intermediate in the pentose phosphate pathway. D-erythrulose is used in some self-tanning cosmetics, generally combined with dihydroxyacetone. 3.7.4 Trioses. The simple sugars that have a 3-carbon backbone are called trioses. There are 2 important trioses, one is an aldotriose, namely glyceraldehyde, while the other is ketotriose, namely dihydroxyacetone. 3.7.4.1 Glyceraldehyde. Glyceraldehyde is a triose monosaccharide with the chemical formula C3H6O3. It is the simplest of all common aldoses. Glyceraldehyde has one chiral center, so it occurs in two enantiomers, one of the two stereoisomers that are mirror images of each other, with opposite optical rotation. Figure 3.14 Structure of trioses, D-glyceraldehyde, left, and dihydroxyacetone, right. 3.7.4.2 Dihydroxyacetone. Dihydroxyacetone is the simplest of all ketoses. It is an isomer of glyceraldehyde and is formally called glycerone. It has no asymmetric carbon atom. Characteristic features of trioses. Dihydroxyacetone has an important role in the metabolism of carbohydrates. Dihydroxyacetone is used topically to darken the skin. Glyceraldehyde 3-phosphate, a derivative of glyceraldehyde, is an intermediate in the metabolism of glucose, glycolysis. Glyceraldehyde is used as the reference molecule for handedness in amino acids, carbohydrates, etc. 3.8 Chemical reactions of monosaccharides. The chemical reactions of monosaccharides are due to the presence of aldehyde, ketone, or hydroxyl groups. Following are the important chemical reactions of monosaccharides. 3.8.1 Reaction with phenylhydrazine to form ozazone. Ozazone is the yellowish crystal produced by heating the sugar solution with phenylhydrazine. Those sugars which possess free aldehyde or ketone groups can produce ozazone crystals. When the reducing carbohydrates are treated with phenylhydrazine at 100 C and pH 4.3, a series of reactions take place resulting in the formation of ozazone of the respective carbohydrate. The general reaction is glucose plus 3-phenylhydrazine glucosazone plus C6H5NH2 plus NH3 plus 2H2O. The reaction involves only the first and second carbon atoms of the reducing carbohydrates. Since glucose and fructose differ only concerning the first and the second carbon atoms, they form the same ozazone. Mannose will also from the same ozazone. Galactose differs from glucose at carbon number 4, therefore, its crystal shape is different. Sucrose and starch do not form ozazone, because they are non-reducing substances. They can form crystals only if they are first hydrolyzed into their components. Figure 3.15 Formation of ozazone. Generally, monosaccharides give crystals on heating and all disaccharides give crystals on cooling. The ozazone of different carbohydrates differ in their time of formation, solubility, melting point, and crystalline structure. Yellow crystals will appear in the tubes containing glucose and fructose within 10 minutes of heating. Galactose gives crystals within 20 minutes of heating. Maltose gives crystals within 10 to 15 minutes of cooling. Lactose gives crystals within 20 to 25 minutes of cooling. Table 3.2 The characteristic features of ozazones of glucose, mannose, fructose, maltose, and lactose. Carbohydrate. Ozazone compound. Solubility in boiling water. Shape of ozazone crystals. Glucose. Glucosazone. Insoluble. Bundle of grass or needle-shaped. Mannose. Monosazone. Insoluble. Bundle of grass or needle-shaped. Fructose. Fructosazone. Insoluble. Bundle of grass or needle-shaped. Galactose. Galactosazone. Insoluble. Rod-shaped. Maltose. Maltosazone. Soluble. Sunflower-shaped. Lactose. Lactosazone. Soluble. Puffball-shaped. Galactosazone, lactosazone, maltosazone. Glucosazone. Figure 3.16 Shape of different ozazone crystals. 3.8.2 Reduction to form sugar alcohols. Both aldoses and ketoses may be reduced to the corresponding polyhydroxy alcohols. Sugar alcohols are formed by the conversion of the aldehydic or ketonic group into the alcoholic group. Fructose may produce two kinds of alcohols, i.e., desorbitol and demannitol. It is because of the appearance of a new asymmetric carbon atom. Table 3.3 Alcohol derivatives of monosaccharides. S.No. Monosaccharide. Alcohol derivative. 1. Glucose. Sorbitol. 2. Fructose. Sorbitol and mannitol. 3. Galactose. Galactitol, dulcitol. 4. Mannose. Mannitol. 5. Ribose. Ribitol. 6. Glyceraldehyde. Glycerol. 7. Dihydroxyacetone. Glycerol. Clinical aspect: mannitol is used as a diuretic to treat patients with cerebral edema, as it acts as an osmotic diuretic to decrease the water content of the body by increasing the urinary outflow and thus bring about a decrease in brain swelling. The early formation of cataracts in the lens of diabetic patients is due to the deposition of sorbitol. Sugar alcohol such as sorbitol is commonly used as a commercial sweetener in sugar-free diets. They are not easily absorbed and do not raise blood glucose levels as much, have fewer gastrointestinal side effects if used in limited amounts. 3.8.3 Oxidation to form sugar acids. Oxidation of aldoses with bromine water converts the first aldehyde group to the carboxylic group and thus forming the corresponding acid. Br2 plus H2O rightward arrow HOBr plus HBr. Aldoses, for example, glucose may oxidize under proper conditions to form gluconic acid, glucuronic acid and glucuric acid. I formation of gluconic acid. Reaction of glucose with bromine water brings about oxidation at C1 to form gluconic acid. 2. Formation of glucuronic acid. Oxidation of glucose at C6 produces glucuronic acid. 3. Formation of glucuric acid. Oxidation of aldose, glucose, with strong acids example HNO3 converts both aldehydic and primary alcoholic group to the carboxylic group forming dibasic sugar acid, i.e., acid group on first and the last carbon atom. D-glucose plus NO3 D-glucosaccharic acid, glucuric acid. Figure 3.17 Structure of sugar acids. Clinical aspect: in the body glucuronic acid is used in the process of detoxification of camphor. Benzoic acid, steroid hormones, bilirubin, etc. 3.8.4 Action of alkalis. Sugars behave as very weak acids and form salts in a high alkaline medium. In alkaline solutions, all sugars having free aldehyde or ketone group can metabolize to enediol. Enediols are those which have two hydroxyl groups attached to the double-bonded carbon system. Figure 3.18 Formation of enediols. When reducing sugars are placed in an alkaline solution, they are tautomerized, isomers that differ only in the distribution of electrons, and become chemically more powerful reducing agents. Sugar enediols are strong reducing agents. All sugars containing the free sugar group undergo enolization when placed in an alkaline solution. In strong alkalis, enediols are very unstable and reactive. Carbohydrates such as sucrose which do not contain a free sugar group are not enolized by alkali and are relatively stable in alkaline solution. Glucose can be changed into fructose and mannose on reaction with dilute bases at low temperatures. It occurs due to the rearrangement of groups around anomeric carbon atoms. In this way, these three hexoses are interchangeable with one another. The action of concentrated bases may bring about polymerization and fragmentation. 3.8.5 Action of acids. When sugars are heated with strong acids, they are hydrolyzed into monosaccharides. These monosaccharides undergo dehydration, removal of a water molecule, by the acids. Depending on the type of monosaccharide, these dehydrated monosaccharides are called furfuryl derivatives. 3H2O C5H10O5 C5H4O2 Pentose furfuryl 3H2O C6H12O6 C6H6O3 Hexose hydroxymetal furfuryl. 3.8.6 Formation of glycosides. Glycosidic bond is a type of covalent bond formed by anomeric hydroxyl of sugar with the hydroxyl group of another sugar or non-carbohydrate residue called a glycon. Glycosides are the compounds having monosaccharides linked by acetal linkage of the glycosidic bond. Water molecule is released during the formation of a glycosidic bond. As it involves an anomeric carbon atom, therefore these glycosides may be alpha or beta types. RO plus HOR ROR plus H2O. E.g., methanol reacts with the anomeric O on glucose to form methyl glucoside, methyl glucopyranose. Figure 3.19 Formation of glycosidic linkage in methyl alpha-D-glucopyranose, methyl glucose. There are two types of glycosidic bonds, i.e., O and N types, depending upon the involvement of a glycon to attach with sugar through its O group or NH2 group. The aglycon may be methyl alcohol, sterol, phenol, purine, pyrimidine, a protein, or lipid. Clinical aspect: glycosides are an important group of drugs. Some of them have action on the heart as derivatives of digitalis and strophanthus, having steroid as an aglycon, including fluorazine, salicin, amygdalin, and saponin. Glycosidic bonds are frequently found in Di, oligo, and polysaccharides. 3.8.7 Formation of iodo-compounds. Iodo-compounds are formed when an aldose, for example glucose, is heated with concentrated high. Glucose loses all of its oxygen and changed into iodohexane. C6H12O6, glucose, plus CONC high C6H13I, iodohexane. The iodohexane is a straight-chain compound. 3.8.8 Formation of esters. The formation of esters by sugars is due to the presence of the O group. It can react with anhydride and chlorides of many organic and inorganic acids to form esters. The hydroxyl group of sugars may be esterified with phosphates, acetates, and propionates to give various types of esters. The sugar phosphates, esters of sugars with phosphoric acid, are of great biological importance. Nucleoproteins of cells also contain phosphates of ribose or deoxyribose as esters in combination with various nitrogenous bases. 3.8.9 Formation of amino sugars. Amino sugars are produced when the hydroxyl group of monosaccharides is replaced by an amino group, NH2. In most instances the amino sugars are N-acetylated, i.e., an acetyl group. O. CH3C is attached to the nitrogen atom of the NH2 group. Important types include glycosamines, glycamine, such as D-glucosamine, D-galactosamine, and D-manosamine, all of which are derived from hexoses and occur in nature. They are also known as hexosamines. In all of them the NH2 group is attached to carbon number 2. Glucosamine is a constituent of hyaluronic acid. It also exists as a chief organic constituent of the cell wall of fungi. In the shells of crabs and lobsters, it is present as chitin. Chemically, chitin is a repeating unit of N-acetylated glucosamine called chitosamine. Galactosamine, also called chondrosamine, is present in bones, cartilages, tendons and heart valves as chondroitin sulfate. Manosamine is an important constituent of mucoproteins. Another type is glycosylamine, in which the O group of a anomeric carbon atom is replaced by the NH2 group. Ribosylamine is an example of glycosylamine, a derivative of which is involved in purine biosynthesis. Amino sugars also occur in many antibiotics such as erythromycin and carbomycin. Figure 3.20 Structure of amino sugars, glucosamine, left, and N-acetylglucosamine, right. March 8, 10 Formation of deoxy sugars. Deoxy sugars are produced after the loss of oxygen of the O group, leaving behind the hydrogen only. Deoxy sugars of biological importance are 2-deoxy-D-ribose, generally called 2-deoxyribose or only deoxyribose, and 6-deoxy-L-galactose, L-fucose. Deoxyribose is an important constituent of deoxyribonucleic acid, DNA. It gives most of the sugar reaction except the formation of ozazone. L-fucose is present in milk, blood group substances and some bacterial polysaccharides. Another important deoxy sugar is 2-deoxyglucose, which is extensively used in research to see the local effect of glucose metabolism in the tissues. March 8, 11 Formation of amino sugar acids. Important amino sugar acids are neurominic acid and muramic acid. Neurominic acid is produced by aldol condensation of pyruvic acid and D-manosamine. It is unstable and found as acylated derivatives known as sialic acid, i.e., N-acetyl-neurominic acid, NANA. It is an important constituent of mucopolysaccharides and gangliosides. Muramic acid is a product of condensation of lactic acid and D-glucosamine. It is present in many bacterial cell walls. March 8, 12 Fermentation. The reaction of fermentation is usually represented by the following equation. C6H12O62, C2H50, plus 2CO2. However, the reaction is not complete, because many other products are also produced. Some of the hexose sugars are converted to ethyl alcohol and CO2 bisymase, the group of enzymes present in yeast. Glucose, fructose, and mannose are readily fermented by the ordinary strain of baker's yeast, but galactose is fermented to a negligible extent or not at all. Yeast can also ferment disaccharides such as sucrose and maltose. The disaccharides are fermented in two stages. In the first stage, monosaccharides are liberated, and in the second stage, the monosaccharides are fermented. Certain bacteria, Lactobacillus acidophilus and Lactobacillus bulgaricus, contain enzymes that can ferment lactose to form lactic acid, which brings about the souring of milk. Exercise questions. MCQ 1. Which of the following monosaccharides is unable to form D and L isomers? Dihydroxyacetone, erythrose, glyceraldehyde, ribose, ribulose. MCQ 2. Which of the following substances is released during glycosidic bond formation? Ammonia, carbon dioxide, hydrogen, oxygen, water. MCQ 3. Which of the following metabolic processes produce glucose from non-carbohydrate sources? Gluconeogenesis, glycogenolysis, glycogenesis, glycolysis. Please write answers in the comments. End of Part 1 of 2 of Chapter 3. Thank you.